Okay. Good morning, everybody. My name is Wim van der Poel of Wageningen University. I'm professor of emerging and zoonotic viruses, and I've been asked to lead you through this first session together with my co-chair, Julia Gallo. So welcome, everybody, and also thanks to Tatjana and colleagues for hosting us. I think Ljubljana is a very nice place, and I'm happy to be here. I look forward to a very nice meeting, and I will start doing that or helping that with chairing the first session. The first speaker will be Diana Bell of the University of East Anglia. Diana is a conservation biologist, and her talk will be Beyond SARS-CoV-2. Diana, please take the floor and start your talk.
Thank you very much. So I think this is working. So good morning, and thank you for inviting me to the beautiful Slovenia to talk today. I'm going to be mentioning fur farming, and I just thought I'd start with this slide, and you can see how the mink in a Swedish mink farm mutilate one another and indeed kill and eat one another. Now, to look at beyond SARS, we've got to start by looking back. So let's look back to SARS-CoV-1, the first severe infectious disease of the 21st century, index case in Guangdong, 2002, 8,500 cases and 11% mortality. And the cause was a new coronavirus. And there were early indications that this had animal origins because a number of the early human cases were people that lived near or around the markets or people that worked in the wildlife markets. And Iguan went down from the University of Hong Kong and tested a suite of animals in the market and found SARS-like coronaviruses in 3 small carnivore species, a masked palm civet, a raccoon dog, and a Chinese ferret badger. Now, these represent 3 different families of carnivores. We've got the viverids, the canids, and the mustelids. And the initial focus for control in China remained on culling farmed civets. They're farmed for their meat, their alleged medicinal qualities, and civet coffee. But we were working just across the border. I just happened to have a PhD student working across the border in Vietnam on civets, civet ecology, civet conservation, and wildlife trade. And we said at the time, actually, we think you're focusing in the wrong place. You should be looking at the wildlife trade. Scott Robertson was the PhD student. This just shows some of the network of trade routes in Laos and neighboring Vietnam for domestic consumption and trafficking to China of the wildlife. And so we were wondering whether the animal reservoir for SARS extended outside China in one of these countries that had the wildlife trade and was sending it to China. And in these countries, you've got similar wildlife markets mixing the same species and their pathogens and parasites. And we also thought that it was important to screen possible host species at different stages in the wildlife trade route system. So is it present in the wild in these countries, in the wild populations, or do they pick it up whilst they're in the backs of vans and other holding facilities en route to their destination? This is Scott, and indeed he's He works at WCS now, and he and his colleagues there published a paper a couple of years ago now showing that coronavirus transmission risk did increase along that wildlife supply chain to human consumption from the wild to destination. We also thought it was important to look at related taxa. There are 12 other species of mustelids in Vietnam alone, 12 other species of viverids, and if these really were the sources, then it was important to look at these related species. So we were invited to talk at the Royal Society Lessons from SARS meeting in London in 2004. That's the publication. Around that time, the wildlife, the people were testing the wildlife market traders and the civet farmers for SARS antibodies. And interestingly, the civet farmers tested negative for SARS antibodies, whereas the wildlife traders tested positive. So we were right in our suggestions. We also talked about the conservation implications of this situation, which was a severe depletion of wildlife in an Indo-Burma hotspot, which means high numbers of species, high threat, and a high number of endemics. And indeed, in this region, wildlife trade is a greater threat to biodiversity than deforestation. And so we concluded that a major lesson from this work was that the roots of new zoonotic diseases may lie in the parallel biodiversity crisis of species loss. And we call for a One Health interdisciplinary research approach. We also looked at the causes of the development of the trade. And essentially, in the last 30 years in Vietnam and Laos PDR, there's been a shift from subsistence hunting to feed the family, so local consumption of wildlife, to selling that wildlife into the illegal wildlife trade system. The hunters get more money. It's sold as as live animals, unlike Africa and the bushmeat trade where they're dead animals. They're more expensive than domestic animals because they're seen as healthier. They're seen as a luxury and a medicinal item. And with increased affluence, you see that it's developed into this global international trade chain with links with other organized crime like drug smuggling. So the consequence is the depletion of wildlife, but also it's a perfect storm for the emergence of new zoonotic diseases because of that shift from local consumption to mixing of species and their pathogens that wouldn't normally mix in the wild, and then transporting them over vast distances together. So when COVID hit in 2020, I wrote an article for The Conversation entitled The Lessons We Still Haven't Learned from SARS. And the early pro-meds were suggesting that the environmental specimens from the wildlife section of the Wuhan market were 100% identical to SARS-CoV-2. Like many others, I called for a ban on wildlife trade and on wildlife farming. And I emphasized that it was crucial that other countries involved in that wildlife trade chain, such as Indonesia, Thailand, and so on, follow suit, as these supply the same species fueled by demand from China, but also domestic consumption. These are rats waiting to be killed and eaten in the markets, and these are domestic dogs on the right-hand side, just arriving at the market. So instead of a decline in wildlife trade since SARS-CoV-1, there's actually been a global expansion, a rapid global expansion. It's now not only direct trade, but it's online. And species are appearing in the trade in China and Southeast Asia from vast distances away. So African and South American fauna are turning up in the trade. So African pangolins, for example, they weren't in the trade in 2003, but they certainly are now. All the pangolins are threatened with extinction. And South American jaguar and African lion bone are replacing Asian tiger in traditional Chinese medicine. So you've got this suite of animals being shifted to China. So it's expanded. And even during the lockdowns, even during the pandemic, there were international confiscations at borders. There was a big confiscation at Mexico airport in the middle of lockdown of animals from South America that were en route to China. So China did introduce a ban. It introduced it in February 2020, and it prohibited the hunting, trade, transportation, or consumption of all terrestrial wildlife species, whether wild source or captive bred. But there were exemptions. First of all, aquatic wildlife wasn't included, so otters would be regarded as aquatic wildlife, for example. And current legislation still legalizes the trade for traditional Chinese medicine, fur farming, pet farming, research and biomedical research purposes. But there is a ban in China. But unfortunately, in the adjacent countries, there have been zero legislative changes. So it's still legal. Now, I'm particularly worried about wildlife farming, which has been expanding over the last decade. We first studied it in Vietnam in 2010 in porcupines. And what we discovered was that these porcupines are being laundered from the wild. So they're not these farms aren't breeding the porcupines. They're buying them from hunters and then selling them on to other farms. So they're not successfully breeding them under these awful conditions. So even previously common species like porcupines are now on the endangered list. And that's just one example. Now, during the pandemic, Andrew Cunningham and I thought it would be a good idea to look at horseshoe bats in the UK. We've got two species, lesser horseshoes and greater horseshoes, to see whether we got any SARS-like sarbecoviruses in our bats. And we were fortunate enough to collaborate with Miles Carroll, Jack Murphy, and the team at Porton Down and Oxford University, who did the genetic analysis for us. And what this showed was that, well, I had a student, I had an undergraduate actually, who was working for a bat consultancy, and so had the licenses to collect the bats. And we then handed them over to Miles and his team. And 1 out of 53 of these bats tested positive for a new SARS-like coronavirus, RHGB01. You can see in these phylogenetic trees, it's clustering, it's right at the top, and it's clustering with a sequence from a Blasius bat from Bulgaria, collected in 2008. So the lesson here was you didn't have to go to Southeast Asia to find sarbecoviruses in bats. And there've been a couple of recent papers doing more work on UK bats. This study focused on horseshoes again, APA's paper, and they studied, or they actually collected a very good sample of lessers and graters, and from a wide variety of roost sites. And what they found was that there was widespread sarbecovirus infection in the lesser horseshoe bats. 44 and 56% of the samples were testing positive. They only sequenced a few, but you can see them in red clustering with hours at the top. So we get up to 6 new sarbecoviruses. They didn't find any in the graters. Then a paper by Tan et al. They looked at a wide range of UK bat species. There are about 17 bat species in the UK. If you just look at the bottom right-hand phylogenetic tree, even though they had small sample sizes, they still found 2 more sarbecoviruses, 7 and 8, in the lesser horseshoe bats, and they also found 2 in the greater horseshoe bats. And worryingly, 1 in the lesser horseshoe bats, GB7, they suggested could bind with humanase 2 cell and use it for cell entry in vitro. Now I want to change tack slightly. It is related, but to some other work that we've been doing, and it's something I think we should be thinking about beyond SARS, and that's some neglected pathogen vectors, namely mites. These are the smallest arthropod vectors, over 3,000 known species of trombiculid mites, wide distribution, and associated with a variety of hosts. And they've got a really interesting life cycle. They're only parasitic in the larval form, which is, if I can get this working. No, I can't. Oh, yes, I can. That's the larval parasitic form. And the pathogens that they carry, it's been showed, pass transstadally to the nymphs and to the adults, and then transovarally from the mother to the eggs, and then hatch out in the larvae. And the reason that they've been neglected, I think, is because they're so tiny. A big one is 0.2 millimeters in diameter, and they're difficult to study. But this is on the right-hand side. This is a trombiculid, near trombicular autumnalis, the harvest mite from the UK. And several other authors have been asking for more work on mites. There was a paper decrying the lack of work in the States by Chen et al. And the last paper listed there asks whether they carry other viruses apart from the hantaviruses that they're known to carry. So these are the pathogens that mites have been shown to carry so far. 3 hantaviruses, 4 species of Borrelia, so Lyme disease, Orienta, I can't pronounce that, but it's scrub typhus, and then a large number of rickettsias, but all these others, including Mycobacterium. And we were interested in the viruses because we've been working on. I just mentioned this first of all, that I found a paper which actually reported co-infection of scrub typhus and SARS-CoV-2 in a case in Nepal. And this paper was saying that in countries like Nepal, you had to test for other acute diseases like dengue, malaria, and so on. And so it was a greater resource burden. But to me, an interesting question was, did the mites become co-infected with SARS-CoV-2 when they bit the victim? And how many other co-infections with these other tropical diseases have been missed? So we've been studying myxoma virus, myxoma epidemiology in a 30-year study of the European wild rabbit at UEA. And this is a vector-borne virus, and it's alleged that the vector is the flea in the UK, mosquitoes on the continent. And I was fascinated by the highly predictable timing of the outbreaks. And you can see it tends to be in July that they start in this 11-year period, for example, and the epidemic lasts about 6 weeks. Myxoma virus, of course, we all know the story. The natural host is the silver largus cottontail rabbits in the Americas, and it causes a benign lesion at the base of their ears. But that virus, when transferred to a different genus, the European rabbit, Eryctolagus, causes lesions around the eyes, the mouth, base of the ears, anogenital area, and the feet. So we were collecting mosquitoes, fleas, and mites, because I wonder whether there was a mosquito perhaps that emerged at that time of year. Because if you look at this, in the blue is the flea load, and you can see that the flea load is at its lowest during the green myxomatosis epidemics, whereas the mite load matches perfectly with the myxoma outbreaks. And so I had a PhD student that not only looked at the number of harvest mites on rabbits that we were trapping regularly and inspecting. They were all individually tagged for identification. So we looked at the location. We also collected emergent unfed questing mites, which just emerged from the soil and were hanging around on the vegetation waiting for a mammalian or avian host to bite. I sent the student with the fleas, the mites, and the mosquitoes to Canberra, to Peter Kerr's lab, because he was the myxoma expert at the time. What we found was that the lesions were, the mites were in all the places that you got the lesions, so around the eyes, ears, anogenital area, and feet. And you could catch, they occur in mite islands. So you can get a big cluster and then nothing. And when we got a big cluster, we could catch up to 800 mites on a 1 meter square piece of cloth in 10 minutes. But most excitingly, 50% of the pools of 10 harvest mites that we sent to Australia with my PhD student tested positive for myxoma virus. So these larval mites were carrying myxoma virus. And so we can add leporopox viruses to the list of pathogens that mites are carrying. And I don't think you need to be a rocket scientist to see that the other pox viruses that I would suggest that mites might carry are sheep pox, goat pox, cow pox, because you've got the same lesions in exactly the same places where the skin is thin, around the nose, around the eyes, around the ears, around the udders, all places that are going to have maximum contact with the questing mites on the vegetation. So, like all of us here, I'm asked what the next pandemic's going to be. And my response at the moment is that we've already got a pandemic in biodiversity. which is H5N1. We've all seen how it's travelled to mammals and to bird species in South America, in North America, across Europe, we actually worked on this in 2005 because H5N1 killed some of our critically endangered Austin's palm civets that you can see on the right. This is work we did with Malik Paris in Hong Kong, and also Peter Horby. And what we included in that publication was a list of all the species of birds and mammals that were known to die from H5N1. And it was extensive then, but we've just repeated that, and it's vast now. And we suggested then that anything could die from H5N1 or that particular strain, but unfortunately it seems to be true. Predictions that this will be the next human pandemic again, because it killed people in the early 2000s. And this is my last slide. I just want to say that I think that in addition to banning wildlife trade markets and wildlife farms, we should be banning fire farms. I agree with these authors here, Peacock et al., in a recent paper. We've seen how good mink are at spreading both H5N1 and SARS-CoV-2. This is a raccoon dog, and ironically, it's cheaper to produce fur from a live raccoon dog than it is to make fake fur, which gives you some idea of the conditions that these animals are kept in. And on the right-hand side, we've got a shipment of Danish mink being shipped to China for processing. Denmark, of course, banned, they culled all their mink in 2020, but they've started farming again. I'd also, secondly, like you to consider when you see that a disease is rodent-borne with direct contact with rodents, for example, allegedly, or with a feces or urine, or even tick-borne, it might be mites. But whatever they do, we need a One Health interdisciplinary research approach to this research. And I'd just like to acknowledge all these collaborators without whom this work wouldn't have been possible, including ProMed. Thank you very much.
Thank you very much, Diana, for this nice talk. Are there any questions? Sorry. Okay, go ahead.
No, fascinating. I know some of your work, but putting it in that context together was quite incredible and obviously very scary, and also reveals the stupidity
Yes.
behind it as well. On the mite question, you mentioned that the viruses have been found in the bacteria as well.
Yes.
What are your plans for looking further? I mean, the viruses that they found, were they targeted in their sequencing approach, or was it an Illumina sort of deep sequencing so they could find anything? And obviously, it depends what species you take the mites from and from what location. So what are your plans for furthering that work?
Well, so I've just become an emeritus professor at my university, but it's not going to stop me from looking at questing mites. I've got a large supply of mites in tubes for anybody that wants to collaborate on this. You know, indeed, you know, I wherever I see a new virus that I haven't heard of that's said to be associated with a rodent, I just think it's might. So I think we need to do a kind of targeted and I mean I think maybe bovine TB would be worth looking at, really worth looking at, because it's burrowing bad badgers. The virus might come from the cattle, or the bacteria might come from the cattle. Sorry, and then in fact get into this cycle. But I just think it's a very clever. Very clever.
Well, no, certainly give us some ideas of the work we're doing in West Africa. We didn't think of mites, but we should be looking at those.
Yeah, absolutely. I might look at bat mites.
We'll do.
Yes, yes.
Okay, thank you. Are there any other questions? Yeah.
Really interesting. With the myxoma positive mites, are you saying then that you think that the myxoma is making it through the whole life cycle of the mite?
Absolutely, yes. Yes.
Is there evidence for that, or is that just what must be happening to explain the data?
Well, so we haven't fulfilled Koch's postulate in terms of actually putting the mites on experimental rabbits, because it's such a horrible disease. I can't personally do that research. But you know, we know with scrub typhus that's what happens. You know, they've followed it through the cycle, and one or two other diseases where they catch it. It's important that you find it in the questing mites, because then it's impossible for them to have bitten another host. They're bright red. The harvest mites are bright red when you catch them, and if they haven't fed, they turn orange when they've fed. So we knew that they hadn't been fed.
Okay, it's just that. I mean, how does it work then to get all the way through the life cycle? Does the virus have to replicate in the mites, or can it sort of remain dormant?
Well, we don't know. Those are all questions that.
Because it's an interesting evolutionary,
yes, I know.
co-evolutionary thing.
I know.
Yeah.
I know.
Thank you.
Could you also play a role in avian influenza, because mites are very prevalent in poultry also?
Yes. Yes.
Okay, there was another question there.
Thank you. How species specific are the serpicoviruses within the bats? Because if one wanted to do experimental work and try to understand permissiveness within bats, and there's a lot of bat diversity, you've got the Debentons, the Pipistrelles, the horseshoes, do you think the evidence here is that it's quite species specific, or do you think it's just we haven't looked hard enough in enough species?
We haven't tested enough species yet. I think it'll be very interesting to see the results. I know Miles's team is doing that research right now, so whether it's species specific or not, I don't know. But there'll be, you know, there are 85 species of horseshoe bats, and they've got a distribution across the old world all the way to Australia. So the potential for sarbecoviruses is huge in terms of potential hosts.
Thank you.
Okay, thank you.